Welcome everyone. I would like to present you my PhD topic, which is antimicrobial therapeutic drug monitoring in critically ill patients by a clinical pharmacist. My name is Eszter Gulyás. I'm working as a pharmacist, and during my residency years, I became interested in intensive care medicine-related clinical pharmacy, and my mission is to promote therapy guidance with drug monitoring in intensive care and to become an expert in the utilization of therapeutic drug monitoring results. I have two ongoing projects, both is related to antibiotics and TDM in critically ill patients. The first is a meta-analysis. Its title is The Impact of Antimicrobial TDM of beta-lactam antibiotics on the Clinical Outcomes in Critically Ill Patients based on PK/PD target attainment. I would like to tell about the background that 70% of the critically ill patients receive antibiotics during their hospitalization. But this patient population is a completely different population to those volunteers or not severely ill patients. So the inter and intra-individual variability regarding the pharmacokinetics is very different. And we cannot be sure that the PK/PD targets are reached. That's why we could imply routine TDM, therapeutic drug monitoring, which is the measurement of drugs concentrations in blood fluids in specific intervals, and not only measuring, but the adjustment of the dosage of drugs is applied. But we don't know the added value and the effects on outcome. So our clinical question was phrased: is TDM-guided beta-lactam antibiotic therapy more effective than standard beta-lactam therapy in critically ill patients? So the intervention is when we apply routine TDM and adjust real-time dosages, whether we do not do this and standard of care therapy is done. You can see here my systematic search, which was conducted in 3 basic databases with a much broader search key. About the flowchart, I had almost 3,000 articles and 7 found to be eligible. I would like to present now my results. First, I would like to start with the hospital mortality. Here we can see relatively low statistical heterogeneity, but I would like to tell that of course, the population was so heterogeneous regarding the diagnosis and the admission of the intensive care. And we can see here 0.87 odds ratio, which doesn't seem to be statistically or clinically relevant. And we could conduct a subgroup for ICU mortality, and the same result has been found with a very broad prediction interval. Of course, mortality is much more complex than to be easily that it does not really depend only on TDM. If I move on to the length of ICU stay, which is the time measured by days between the admission and the discharge of the ward, we can find minus 0.46 mean difference, which means that the TDM patients can be hospitalized on the ICU half day less than the standard of care group. Obviously, it's not really not a critically relevant data, but more promising results. What we found is microbiological eradication, which means that the odds for microbiological eradication was 1.89 times higher as for the TDM group as for the standard of care group. It was not statistically significant, but clinically it was. And the best result we found out is the clinical cure, which seemed to be 2 times, the odds was found 2 times higher as for the TDM group as for the standard of care, and it was found to be statistically significant as well. For summary, I would like to highlight that TDM-guided administration of beta-lactams is associated with improved clinical and microbiological cure. we had some limitations. That small sample size, we could use only small sample size. We assumed that 10 times more sample size will be statistically more significant. We had observational studies, but we had some RCTs. Our population was heterogeneous, and we did the risk of bias, which seemed to be moderate. For conclusion, I would like to highlight that using TDM may be beneficial for increasing clinical and microbiological cure. Well-designed randomized controlled trials are needed, and standardization of protocols and clinical endpoints are essential. For clinical implication, I would phrase like PKPD index-guided dosing facilitates addressing inter- and intra-individual variability of pharmacokinetics in ICU patients, and for research implication, as I said, standardization of protocols, it's a crucial point for PKPD index calculation, and cost effectiveness would be the best what we could connect with TDM. I would like to move on with my second project, which is the clinical evaluation of pharmacokinetic-based guidance of colistin therapy. It is a protocol for clinical validation of the analytical method and the pharmacokinetic investigation. About its background, therapeutic use of colistin. It was discovered in the 1940s, but its use was abandoned in the 1970s due to its narrow therapeutic index. But in the last 2 decades, due to the emergence of gram-negative superbacteria, the resurgence of colistin therapy was shown. But we also have no data how to guide colistin therapy, so TDM is a tool for this. And I would like to highlight that there has not been existing analytical method for colistin TDM in Semmelweis, but to our knowledge, not even in Hungary. So a little bit about its pharmacokinetics. It is a very complex chemical structure. Polymyxin A, B, C, C, D, and E exists, but only B and C is relevant for clinical aspects. And an inactive product, colistin methanesulfonate, is used. It has a wide volume of distribution and 2 to 3 hours half-life, and it eliminates with glomerular filtration. We are following our schedule, pre-planned schedule, and the development of an analytical method is already existing, and the technical validation of the method is in progress. We were able to collect samples from humans for the technical validation during the last week, and as a proof of the existing method, I brought ion chromatogram in spike serum from the laboratory, which my colleagues have done. So after we will be over these steps, we will be able to phrase our Pico, which would be the following: how should colistin be dosed and monitored to improve its therapeutic efficiency in critical care? And the intervention would be that we measure, we administer colistin, and we apply real-time dosage adjustment versus the standard of care therapy. And the clinical implication of this, that we will be able to optimize the colistin therapy. And I would like to finalize my speech with the great news that yesterday we were able to submit our first abstract for a congress with our second project with colistin. And I was told that the way of progress is neither swift nor easy. Thank you for your attention.
Thank you for the presentation. It was really nice. I just wanted to ask that, do you have any information about antibiotic resistance, how this TDM could have effect on this? Have you met any kind of articles, or just it's a theoretical question in a way?
Yes, thank you, Dinesh. It's a very good question. Yes, I was searching for those outcomes as well, resistance, but these 7 articles do not really define the antimicrobial resistance, but it would be, I'm very curious to know, because that would be one benefit and goal for TDM to to be able to measure resistance as well.
Congratulations, of course, it's very important that when you apply a therapy that it has to be efficient. I'm just curious whether, of course, subgroup analysis are always important, but I'm very curious whether you have to monitor all kind of antibiotics, or is there a specific antibiotic that is better to be monitored, or...
Yeah, thank you for the question. This first meta-analysis was about the beta-lactams together. The articles does not really distinguish each kind of drugs, but beta-lactams are a specific group which can be measured together. Of course, subgroups should be done, subgroup analysis, but due to the small sample size, we weren't able to do that.
Thank you, Eszter. Again, a very nice presentation. And my question relates really to clinical outcomes, because in your first project, you put mortality as your first outcome, but what was the primary outcome in the studies you used in your meta-analysis?
Sorry, it was a little bit controversial, but not mortality was the primary. Clinical cure was the primary. Maybe it would be better that...
I mean, both follows a logic. Okay,
yes, yes.
Both strategies follow a logic, but I would rather change it the other way around, because really, I mean, mortality can be a very important outcome, but it is highly unlikely that just one antibiotic therapy has such an important effect on that. And you already mentioned that, by the way. So regarding the clinical outcomes, and that relates to your second project as well, because you mentioned in your first project that microbiological cure and clinical improvement. But how can you measure? What are your aims to measure clinical improvement? Because this is really the importance of, this is why we give antibiotics.
I would like to answer with the article's definition that microbiological clinical cure is defined as the improvement in all clinical symptoms and signs. So it's a little bit not so objective measurement, but this is how all the articles define clinical cure. Maybe it's defined as cessation of antibiotics due to microbiological data and or clinical improvement by symptoms.
It's a very broad term, and especially in your second project, I would specify clinical improvement by vital organ functions, right? Like need for mechanical ventilation, or, which is again a very trendy outcome, is ventilator-free days, vasopressor-free days. It's much better than length of ventilation, because if the patients die in 5 days, they have a shorter day on ventilator, and those who live for 10 days, you see? Yeah. So something like that.
Yes, thank you for the comment.
Thank you, and maybe I can ask a very short question, Eszter, about your first project. So it seems that, it's obvious, TDM can improve the clinical outcome, but is there any explanation, could you find any possible explanation about the way? So how can the TDM increase the overall efficacy of a generally atoxic antibiotic? Maybe without TDM it is underdosed, or something similar pharmacokinetic effect can be in the behind.
Yes, we have done the meta-analysis with beta-lactams, which are seen to be not so toxic. So the toxicity of there is low. So, but I think the key point is here when we have protocols, how to dose them. We have immediate feedback, and we can adjust the dose. I think that is the key point.